Hello, everyone. Thank you for joining us for our May Research Spotlight webinar. We are excited to have you here with us during HD Awareness Month. I'm Dr. Sarah Hernandez, the Director of Research Programs at the Huntington's Disease Foundation. I want to start by thanking everyone who supports the HDF. Because of you, we are able to fund exciting science that is transforming what we know about HD. And I want to thank all of you for being here and tuning in. We are joined today by Dr. Dirk Keene and Dr. Dan Child. Dr. Keene is a professor and the chair of pathology at UC San Diego. He's a board-certified neuropathologist who built and led one of the largest neuropathology programs in the country at the University of Washington, where he also directed the Brain Laboratory and served as associate director of the UW Alzheimer's Disease Research Center. He's a pioneer in human brain biorepository science, including founding the Pacific Northwest Brain Donor Network, and he has dedicated his career to making sure that the extraordinary gift of brain donation translates into real scientific discovery. Dr. Child is a board-certified neuropathologist and faculty member at the University of Washington and co-PI in the Brain Lab. He did his MD and PhD at Penn, where he studied the cardiac effects of Huntington's disease. So HD has been central to his scientific story from the very beginning. His lab uses human tissue and stem cells to model why genetic neurodegenerative diseases like HD affect people so differently, even though they have the same mutation. Together, they'll be sharing an exciting new initiative that they've been building out of the Brain Lab, HD Bridge, a dedicated HD brain biorepository at the University of Washington. The program brings the Brain Lab's gold standard methods to the HD community for the first time, encompassing brain donation recruitment, standardized tissue banking, and researcher access, all under one roof and governed by one of the top neuropathology biorepositories in the country. HD Bridge hasn't officially launched yet, so we are getting a real preview today of something that the field needs and will help advance HD research. Before I pass it over to Dirk, I want to remind you all that you can ask questions in the Q&A box throughout the talk. We'll have about 20 minutes at the end of the session for a Q&A, so make sure you send your questions in as you think of them, and we will try to address as many as we can get to. Also, a reminder that this will be recorded and available on our website and our YouTube channel in about 2 weeks, so if you want to watch it again or if you want to share it with someone, it will be available for that. Thanks again, everyone, for tuning in, and I will now pass it over to Dirk.
Thanks, Sarah. And hi, everybody. It's really a great honor to be able to talk to you today. It's one of the greatest thrills and also one of the most difficult things for a scientist to talk to people like you all, and yet without you, none of this happens. And so I really appreciate you giving me some of your time for us to tell you a little bit about the work that we're trying to do. I'm really thrilled to answer any questions and any kind of follow-up. Sarah and the team can get a hold of us for whatever questions come up. But I just want to start by emphasizing how important brain donation is for science. Everything that we know about what happens in the brain of a person with Huntington's disease pretty much comes from brain donation and from us of the ability to examine brains of people after they've died. It's an incredible gift, the greatest gift I think you can give for science. And it's not just Huntington's disease, it's all other diseases, but importantly, it's also normal brains. We can't understand what's happening in a disease like Huntington's disease if we don't really have a good grasp of what's happening in the normal brain. And the brain is so complicated and so advanced compared to other organ systems and other species. We really need to study the brains from people all across the spectrum. And so this presentation is about some of the research that we're doing and hopefully some of the impact we will make and a little bit about us and how we got here. But what we really want to emphasize is the importance of brain donation from people who have diseases, but also from people who don't, and the work that we go to great lengths that keeps us up at night to try to make sure that we learn as much as possible, not just us, but everybody, that we provide these brain tissues to people around the world doing really brilliant research to understand Huntington's disease. And so I'll start by just sort of explaining my how I got to where I'm at. These are my parents. My dad was born in Iowa. My mom was born in New Mexico. They had very different lives growing up, and then they met in college, fell in love, and got married. and lived very long, healthy, full lives, all the way up until their 80s. That top picture on the right is my mom. She had gotten pretty advanced dementia by that point, but they aged really gracefully, and so my interests from high school even, especially with experience with dementia and those sort of things in my family, has been to understand what happens to people as they age, what happens to the brain as we get older, and then what happens when something goes wrong, when we have some sort of a process that starts the cascade that leads to dementia or whatever we succumb to later in life. My whole goal is to understand how the brain works, how it's built, what happens when things go wrong, and figuring out ways to fix it. And so it all sort of derives from people like you and me, which we all go through these sort of life changes over time, and I want to make that as healthy as possible. But most importantly, these are my kids. Um, I want to help them in the next generation and their kids by learning what has happened in my parents, what's happening in us, so that we can make a really big difference. And so that drives my entire research program. The whole reason we've built the Brain Lab is to really support science that helps us understand how things go wrong and how to fix them, or more importantly, prevent them. So a little bit about me and how I got to where I'm at. I was born in Boulder City, Nevada, home of Hoover Dam. I went to the University of Nevada, Las Vegas, and worked with a guy named Herm Lehman. Dr. Lehman was a neuroscientist, the only neuroscientist at UNLV actually, who studied the tobacco hawk moth. And so I learned about how really cool the nervous system is from Herm, and really realized at that point when I was in college that I wanted to be a neuroscientist. From there, I went to the University of Minnesota, where I did a combined MD and PhD program. And I had the really amazing opportunity to work with Dr. Walter Lowe. Dr. Lowe is a professor in the neurosurgery program department there, and my what I thought I was going to do was become a neurosurgeon and transplant stem cells to treat Alzheimer's disease. I got into Walt's lab, and Walt was studying transplantation, but he told me, there's this really great opportunity to work on Huntington's disease. I have a project for Huntington's disease I'd like you to consider. And at that point, I must admit, I didn't know much about Huntington's disease, but I said, of course, you're my mentor, my idol. I want to do what you say. And that was one of the most transformative decisions in my life. I studied TUDCA, tauroursodeoxycholic acid, which was a bile acid, which was neuroprotective in mice and rats that had models of Huntington's disease, published some high impact papers showing that TUDCA was protective. And I thought that we would be using this to treat patients, before I was done with medical school. The other thing that really happened at Minnesota that was really important was I met Martha Nance, Dr. Nance. Some of you may know Dr. Nance. She's a seminal figure in Huntington's disease and Parkinson's disease, and she really epitomized the clinician's role in helping patients and families with neurodegenerative disease. I can't tell you enough how much I admire Dr. Nance and her interactions and support for patients. And then finally, I met Dr. Brent Clark. He's a neuropathologist, and I realized at that point from Brent that I wanted to be a neuropathologist. So everything sort of converged at the University of Minnesota for me to become a neuropathologist. And I also realized because the treatments that were developing in mice and rats weren't working that well in people, that we really needed to understand human brain before we can go and model it. And so this is a picture of a human brain. Down below at the bottom is a mouse brain. And the reason I point this out is I want to emphasize to people, if we see something happening in the human brain, it's relevant to all of us as people. If we see something happening in a mouse brain that doesn't happen in a human brain, it's interesting and it's important for mice, but it may not be that important or that relevant to what we are dealing with as humanity, as a society, and as patients and families. And so at that point, I really decided I want to really focus on human brain. And so I did. And so I went to um, the University of Washington, where I became a neuropathologist. And I I learned a lot about why we should be studying the human brain, not just because it's relevant. The human brain is amazing. It's composed of 100 billion neurons. It's hard to imagine that. But then each of those neurons can be connected to 10, 50, 100, 1,000 other neurons. And so the complexity is just unmatched in anything that we have biologically. There's over 5,000, sorry, that's a mistyper. There's over 5,000 types of cells in the brain. That's incredibly complicated, right? I talked about relevance, but the human brain is so large and so um, you know, there's so much human brain that when we do have the opportunity to study human brain, we can study human brain in a lot of ways, across a lot of labs, across a lot of time. There are people who donated their brains 20, 30, 40 years ago at the University of Washington, and we still actively share their tissues with brilliant scientists, people way smarter than me, who are doing research around the country and around the world on Huntington's disease and other disorders. And so one brain donation can have an incredible impact, not just for the research that's being done at that place, but for research elsewhere and research over time. And so the human brain is really important. I sort of came to the University of Washington at that point in Seattle. I got to work with Dr. Buster Alvord on the right and Dr. Cheng-Mei Shaw on the left, who were famous neuropathologists and really set the example for the kind of professionalism and academic, you know, um, curiosity that really stimulated me as a neuropathologist. And I really became so enamored with the human brain and studying the human brain in the microscope. I also had the great fortune to meet Dr. Tom Bird, who's spent a career focusing on helping Huntington's disease patients and families as a neurologist. Tom is legendary. He's written a book about his experiences in the Huntington's community with some of his patients that many of you, I'm sure, know about. Tom is amazing. He's very active in working with us to this day. He's been my idol, and I know Dan's idol, and many other people's idol. And so I wanted to really emulate Tom when I got to the University of Washington. And then right when I got there, I started my neuropathology career by having the opportunity to study three Huntington's patients who had received cell transplants. Remember, I went to Minnesota thinking I wanted to do cell transplantation for Alzheimer's disease and ultimately Huntington's disease. And as a neuropathologist studying the first few brains that had ever been seen with transplants, it was an incredible honor. And I realized, you know, the transplanted cells lived for years and years and years in those folks, but they didn't do a very good job connecting to the rest of the brain. So there was a problem with these transplants. And so it clearly wasn't the solution. We really needed to go into the brain, understand the brain better and what's happening in Huntington's disease so that we can engineer the cells within the brain rather than putting new cells in the brain that aren't really designed to connect and regrow in the brain. And so at the University of Washington, I also had the opportunity to work with Tom Montine, who's legendary. He's a professor and chair at Stanford now. Tom instilled in me the importance of quantitative neuropathology. Neuropathology is the study of brain, whether and spinal cord and other parts of the body, the nervous system, usually under the microscope. Tom really instilled in me the idea that we can learn more than just what we see in the microscope. We can measure things and quantify things. We can apply biochemical methods and molecular methods to brain, and we can learn so much more than we can just in a microscope. He didn't devalue the microscopy that we do, but he emphasized how important that was. And I learned a great lesson. And then finally, I met Dr. Ed Lean at the Allen Institute. Ed and I have worked together for 10 or actually probably 12 or 14 years now. And I'll show you at the end of this presentation the impact that working with Ed has made and the big plans we have going forward. But one of the advantages, one of the great things about working with Ed is I got to sort of rethink our biorepository, our brain bank. And that was where the Brain Lab was born. The Brain Lab is really focused on integrated neuropathology and advancing methods and innovation with respect to research neuropathology. We have an amazing team at UW. As I said, I just left to be chair at UC San Diego. The opportunity at UC San Diego is one that enables us to build a network of these world-class brain banks. And I've been really, really fortunate to work with many, many people. A lot of these folks are my trainees who are now faculty members, including this amazing neuropathologist scientist, Dr. Child, who you'll hear from in a little bit. So Dr. Lean, when I started working with him, this is a complicated slide, but basically all this is saying is there's a massive effort by the National Institutes of Health to understand the normal brain from mouse to human. And this is called BICCN. The focus of BICCN, or the Brain Initiative, was to develop new tools, new technologies to study the brain so that we can ultimately build a brain atlas. Think of this like the Human Genome Project. When we were first starting out to understand DNA and sequencing DNA, remember how many of you may not even remember, but when we got the first DNA sequence of a human, it was revolutionary. This is what we're trying to do with the brain and building an atlas of all the cells and their connections and how the brain works in people ultimately. And so with Ed, he was leading efforts at the Allen Institute and a lot of these other things, the other programs, and he came to me and said, you know, the brain tissue that we have available at this right now is inadequate. It's not prepared or preserved in a way that allows us to apply these new tools that we've built, these molecular tools that are really revolutionary. But the problem is brain tissue needs to be preserved and in a form that is amenable to this, which requires us to think about new ways to do this. So we re-envisioned brain donation from start to finish. This is another complicated slide, but basically it shows up on top that we have this tabletop MRI. It's a portable MRI that we can use to get MRI scans of people's brains after they die, which enables us to, we can do it quickly. One of the most important parts of brain donation is getting the brain quickly and preserving it as quickly as possible, because the minute a person passes away, their brain starts to degrade. And then we have that blue stuff in the middle. That's dental cement that we use to stabilize the brain while we're slicing it very thin. It turns out that applying MRI technology, slicing thin and then freezing in a different way, has enabled us to perform amazing new technologies on these brain tissues all across the brain. And so that's led to the first draft of the Human Brain Atlas. This was published a couple of years ago. This is the first draft of the human brain that has been published based on the work that we've been doing. Remember, I said how important it is to understand the normal brain? Well, this is the work that's setting the foundation that will allow us to understand what's happening in Huntington's disease as things go wrong. And so this work is ongoing. We're expecting to publish the full atlas in the next few years. And these methods are now applied across the board at UW in the Brain Lab, will be applied across the board in UCSD, and ultimately in the neuropathology network that we hope to build. And so why do all this? Well, the reason is, as I said, if we understand the normal brain, we can then understand what's happening when something goes wrong. And so we started with Alzheimer's disease. There's a very large Alzheimer's center at the University of Washington. And so Dr. Lean and I, excuse me, and a team of people, a huge team of people, lead what we call the Seattle Alzheimer's Disease Brain Atlas, CAD. What CAD is doing is it's taking brains from people with Alzheimer's disease from before they even have symptoms all the way to the end stages, and trying to see what's different in those people's brains than what happens with us in normal aging through the Brain Initiative and other controls. And so what CAD does is it brings together all these new technologies that I mentioned. It brings together all this new quantitative neuropathology that Dr. Montine espoused. It pulls it all together into one site with some really amazing computational and analysts and lots of AI to start to sort of unravel the mystery of what's happening in Alzheimer's disease early. And so this is just a summary of all the results we've gotten to date. CAD is ongoing, but the first results of the first 5 years of this project have revealed some new cells that we see that are vulnerable in Alzheimer's disease before the cells that we classically think about when we're looking in the microscope. These are called somatostatin neurons. You can see a diagram of these neurons over here based on the Brain Initiative work we've been doing. It also highlights inflammation and reactivity and all kinds of other processes that are happening over the course of Alzheimer's disease. And so what this does is it tells us what's changing early, what's underlying those changes within each of these different cells, like what genes are being expressed, what proteins are being made, what's happening in those early cells. And so the work that we're doing in Alzheimer's disease is really focused on figuring out what's the earliest changes and how do we stop that, or how do we intervene. It's really exciting stuff, and you'll be hearing a lot more about this, I'm sure, over the next few years. And so what this all did, all this stuff coming together, the Brain Initiative work that Ed and I have done, and many, many, many others across many universities, the work that we've been doing in Alzheimer's disease, sort of set the stage for the Allen Foundation. The Allen Institute is actually a number of institutes that are focused on brain, immunology, other things. And so this really stimulated the Allen Foundation to start a new initiative, and it's called the Brain Health Accelerator. I'm super excited about this. The Brain Health Accelerator is going to be transformational. It's just something that's grown out of all the work that we've been doing. And essentially, what the Brain Health Accelerator initiative does is it takes and it focuses on building a knowledge base with all these different applications, these different technologies in cohorts of people with different diseases. They've chosen to focus on Huntington's disease in addition to Parkinson's, ALS, and Alzheimer's disease. And so we're really excited that the opportunity for Brain Health Accelerator will be focused on Huntington's disease as one of the main targets. And so what it does is it combines all these different fancy techniques and technologies that are, really complicated. Basically, all this says is the Brain Health Accelerator is going to build a knowledge base like no other that will combine all these different areas to try to get a comprehensive understanding of what's happening in Huntington's disease. But then what's really gonna stimulate is we're going to use that information to then go and figure out how everything works, to model it in cells and mice and other computer systems with lots and lots of AI, with the goal that we'll identify potential targets, druggable targets, and that this will take those new drugs, those potential therapies, all the way to initial clinical trials, to the point where hopefully the pharmaceutical industry will have enough information about that they'll be willing to invest the big bucks that it takes to actually get a drug through. And so we're really excited. There's some new tools that people can use, not just classical drugs, but gene therapies and lots of other things that we'll be working on in the Brain Health Accelerator. And I'm really excited that I get to run, hopefully with Dan at UW and many others, what's called the Brain Health Accelerator Tissue and Precision Pathology Coordinating Center. That's a lot of words to just say we are going to be focused on uh, working with the foundations, working with the different groups to help people understand how to donate their brain, to help people facilitate donate their brain, to help families participate, to find people who don't have Huntington's disease but who wants to donate their brain, all through this new effort called HD Bridge that Dan has really envisioned and is leading. We're really, really excited about HD Bridge. HD Bridge is going to be the foundation that is going to build up our Huntington's disease portfolio in BHA, in the Brain Health Accelerator. And so with help from you, from the different foundations. HDF has been amazing. Our hope is that we can provide a whole new perspective, a whole new knowledge base that not just us, but people around the world can use to help solve this terrible disease and to really make a difference, to prevent the kinds of things that are happening. If we can't cure from at the DNA level, then to help, prevent the process from progressing so that people with Huntington's disease die from other things, not from Huntington's disease. That's really our ultimate goal with BHA. And so Dan is going to talk about, I think, some of the more exciting scientific components of what we're trying to do, emphasizing how important brain donation is in this process.
Thank you, Dirk. It's always a great honor and also a tough act to follow. Coming speaking after Dirk. All right, so as Dirk said, I'm going to pick up and talk a little bit more about kind of the scientific, not only the scientific aspects of what we learn from brain donation and what we've tried to help support and facilitate, but also some of the more practical aspects of brain donation. How do you get involved with brain donation? What is the process like? What does it look like for a researcher who wants to use human tissue? So like Dirk, I'll kind of start at the beginning. Oh, first I should say I've got no financial relationships or commercial relationships to disclose. So I was born in Denver, Colorado, and from a pretty young age, especially during high school, really became interested in science. I was interested in a lot of subjects, but science was the one that I kept coming back to. And so in order to pursue that more, to dive into it, I went to undergrad at Boston University. And while there, I found myself in the lab of an investigator at Mass General Hospital in the Genetics and Aging Research Unit, Dr. Rudy Tanzi. Dr. Tanzi studies the genetics of Alzheimer's disease. And so this was really my first introduction to what it's like to, I'd had classes talking about the brain and talking about genetics. This was my first experience with the actual hands-on work and studying genetics and studying the brain. One of the things that Dr. Tanzi had done was had collected a lot of samples from lots and lots of different people and then separated them and then extracted the DNA and looked at specific markers to try to find which genes were associated with Alzheimer's disease. Once we had generated this list, he passed it over to one of his trainees and now investigator at MGH, Dr. Kan Zhang, and our job then became to ask, what's the mechanism? Is this actually affecting Alzheimer's disease? Is this actually causing a pathologic change? So the way I kind of saw it was that you get this list of genes and it's sort of like getting the blueprints for something. And then with our work, what we were trying to do was essentially take it apart, the actual object, look into the cell, look into the mechanisms, look into the actual biology going on. And I absolutely loved that and decided that's what I really wanted to pursue for my career. So because of that, I decided I needed to go to a place that had a really strong genetics program. I planned on doing both MD and PhD training. So I wanted to go to a place that had good clinical training and also a really robust genetics research. And so for that, I went to University of Iowa. While there, I met my PhD advisor, Dr. Beverly Davidson, but during my second year there, she was recruited to Children's Hospital of Philadelphia. And I was fortunate enough to move with her and continue my training and finish it at Children's Hospital of Philadelphia and University of Pennsylvania. This move, it had two really critical components that shaped my career. The first, obviously, was getting to work with Dr. Davidson. The second was that it was through this work that I really got to become part of the HD community. And as we all know, the HD when you're doing HD research or doing HD clinical work, it's more than just our siloed specialties. HD really is a community of patients and families and researchers and clinicians and social workers and therapists and all of us together working to fight this disease. And that was really monumental for my career, being a part of this community. And I'll get to how it's shaped my future decisions. But before then, I ended up getting a little sidetracked from the brain. As Sarah mentioned at the beginning, when I was working with Dr. Davidson, I sidetracked into the heart for a brief moment. One of the project that we were working on was looking at the hearts in HD mouse models. And you can see here, this is on the left-hand side, this is a mouse that doesn't have HD, just our wild type mouse. And this is the heart from a mouse that has the HD mutation. It's an HD mouse model. And you can see just by looking at this, the heart is noticeably smaller. And we wanted to figure out why that is. When we also found when you induce a cardiovascular stress, these mice respond considerably differently than their wild type, the wild type mice do. So this is a normal HD, or this is an HD mouse that's been injected with saline. You can see nice red cardiomyocyte fibrils just, this is what a heart should look like. And when we induce cardiovascular stress, you get all of this blue staining. This is all fibrosis, fibrotic tissue. So this is frank cardiomyopathy. And we wanted to know why that is. And we eventually were able to tie it back to a molecule called mTORC1. And we found that when we restore mTORC1 activity in HD mice, it rescues the heart pathology. These mice don't end up getting cardiomyopathy. Now, this was really interesting. As Dirk said, it was really interesting for mice. But one of the major limitations and one of the lingering questions from all of this work is this what we see in people with Huntington disease? And this is where we ran into one of the major limitations, is that we really just could not find any tissue to study the heart in humans. And despite, it took us, we looked for years asking people if they had collected the heart material, and it was really, really difficult to find any. And we weren't able to find any by the time that I finished my PhD and moved on. We have in the interim had a few places crop up that say they do have heart tissue. So it's there, but it's really difficult to find. It's not something that's easily available. And I thought, given the fact that I wanted to stay in the HD community, I wanted to continue working with the community, giving back to the community, I thought this is an area that I, as a budding pathologist, I could fill this niche. I could help the community overcome this obstacle. So for that reason, I decided to pursue training in anatomic pathology and neuropathology. And for that, I came to University of Washington, as Dirk mentioned, and trained under Dirk. The reason I chose Washington was really because of its long-standing history with Huntington disease, because of its exceptional brain bank. As Dirk mentioned, the University of Washington has been accepting Huntington disease brain donations for decades. The first donation that we accepted, at least in the bank as it currently stands, was by Dr. Tom Bird in 1963. So it's been more than 6 decades of collecting Huntington disease brains. We now have 106 with CAG repeat mutations ranging from 38 to 160. Given the fact that we have this strong community and the fact that we have been doing this for so long, we're also in a unique position in that we have worked with lots of families, so that we've had donations from parents and children as well as siblings. Dirk also, when he was here, started the Pacific Northwest Brain Donation Network, which helps us to recruit match control donors, which is so these are individuals without HD who aren't necessarily thinking about donating their brain because they're not involved with the clinic. But through this network, we're able to collect these and have access to these. That becomes a really powerful source of tissue to help support scientific studies. At the last year, so another aspect, in addition to just collecting the tissue, we're also very active in supporting research studies. We deliver tissue to researchers worldwide. Last year, we had more than 10,000 samples, not just Huntington disease, across the spectrum of disease and healthy tissue. We get about 1 tissue request per day. So it's a really active biorepository, with the ideal of being, that we are this bridge between the community of individuals who have the disease and families who are dealing with the disease and the research teams, to connect the two and enable this human-focused study of Huntington disease. Now, as I mentioned, I chose UW because of the fact that because of how exceptional the biorepository is. And I figured, this is probably if all of you are like me, this is a question that keeps you up at night. I'm sure all of you have had late nights where you think, what really makes a brain biorepository exceptional? It may just be me, but for the sake of talking about this and going into it, these are all subjective, but this is really the mission of what we're trying to build. This is how we're trying to build this. This is our ideal, and it's something that we're working towards. So first of all, a brain biorepository is at its core a partnership with donors and families. So we need straightforward consent processes that minimize jargon and technical language. We need to have active communication. The families of our donors become part of our community, and we owe it to them to keep them informed, to bring them in, to help them know that we are being the way that we are stewarding their loved one's tissue is responsible, and that it's contributing to these discoveries. As Dirk mentioned, having excellent quality tissue is an absolute must. Part of that is having a really short PMI or postmortem interval. So being able to accept the donation, the brain donation, shortly after an individual dies is really critical to maintaining the tissue quality and preventing degradation of that tissue. In addition to that, maximizing our preservation methods, and then and on top of that, having reproducibility. Not just so when you have multiple samples, knowing that from one sample to one brain to another, the quality is going to be equivalent, but also knowing other details. For example, as Dirk mentioned, there's more than 5,000 cell types in the human brain. So even though a lot of the areas of the brain may look the same, we look at a brain and say, oh, this all looks like the outside, the outer surface. From one region to the next may be entirely different. So being able to know exactly where a sample that is coming from, exactly what part of the brain specifically, is really critical to designing robust research studies. As part of this reproducibility, these brains need to be characterized thoroughly. One of the challenges is working with human brains is that none of us live our lives in a lab or in a vacuum. We all experience we all live human lives that over the course of decades, and as a result having all sorts of injuries and experiences and age-related changes. And so knowing, having a thorough characterization of these really helps. It's really essential to creating a good cohort for a study. Additionally, having accessible de-identified data that's easy to get to and that we can look at and analyze is part of that characterization. And then finally, or not finally, but also as much supporting material as we can will really accelerate what we can learn from these brains. So having ample clinical data, having biofluids or biomarker data, having imaging data, both antemortem, so MRIs, CT scans, both before death and after death. And finally, as much genetic information as we're able to collect that will enable us to design studies, ask specific questions. And then finally, as I mentioned, being able to have access to appropriately matched tissue that will serve as a control so that we know what we're looking at. We know what's being caused by a specific disease, such as HD, as opposed to just age-related processes or injury, physical injuries, trauma, that sort of thing. So where are brain biorepositories located in the US and Canada? So that when you look at this map is, I'm going to start off by saying this is by no means a comprehensive map. This is a map I took from the NIA's website. This is a map of all of the Alzheimer's disease research centers in the United States. Alzheimer's disease research centers are NIH-funded centers of excellence. And as part of these often have really robust biorepositories. So this is just an easy way to kind of say this is a good, this is a starting point for where biorepositories are. There's lots of excellent biorepositories that aren't associated with ADRCs. But the reason I show this map is because a lot of HD brain donations currently in the United States take place through ADRCs. The HD brain donations leverage the existing infrastructure and funding, in part because there is currently no specific dedicated HD biorepository in the United States. I should mention and give credit to University of British Columbia, who has one of the few HD biorepositories, and that serves Canada. But in the United States, really, we have biorepositories that have robust or large HD programs, but nothing that's specifically dedicated to Huntington disease. And as you can see, the little brain icons I put here represent sites that do have a large that have a focus on HD collections, but like I said, nothing that's specifically dedicated, that was built to serve the HD community, to evolve to the needs of the community. That's not to say that if you live outside of these areas, there's no place to for brain donation to occur. The NIH has a biobank called the Neurobiobank that facilitates nationwide donations. But one of the systemic issues that we have is that despite the fact that there's all these biorepositories, we don't typically talk to each other, or we do talk to each other, but everybody does things slightly different. No matter, depending on where you go, you'll get tissue that's preserved in a different way. You'll get different amounts of clinical data. You'll get different amounts of, different specificity for brain region. So there's really not a standardized protocol, and it makes it really difficult when you're trying to study a disease like Huntington disease. It's an uncommon disease. And so often you need to pool samples from numerous biorepositories, and there's differences between what one biorepository and the next, and it makes it really difficult to compare those. And that's really the problem that HD Bridge is trying to address, is trying to make a federated network of biorepositories. So making a network, a system where researchers can go to and get tissue from multiple sites that has excellent quality, has harmonized methods, harmonized data collection, so that they're able to develop and design appropriately powered studies to answer really tough questions, and also enable brain donation regardless of where a donor may live. At this point, I have this slide in here to say, you're all tuning into a presentation on brain donation. So I'm going to show some pictures of brains. Dirk has already shown a few of these, but just to give you a heads up that that's what's coming. So what is it? As I said, everyone does something a little differently. What is it that makes Seattle unique? This is a slide that Dirk's already shown, so I'm not going to spend too much time on it. But the way that we collect brains is we're really trying to maximize this donation and enable us to use this brain and study this brain for years to come. So this involves sectioning it and freezing it in a very specific manner that preserves it in an optimal way for the most cutting-edge molecular studies. It involves multiple rounds of MRI that allows us to do numerous things with it. I'm going to show you some of these here. We do MRI in every single brain. That's more than 1,000 brains in our bank, and a lot of Huntington disease brains. This allows us to get correlation between brains, the antemortem imaging. So we're able to see over the course of an individual's life, if they've had brain imaging, how their brain has changed up to the point of death. We're also able to see things on imaging that you're not able to see grossly. So here, if you look at this area of the brain here, there's nothing that necessarily jumps out at me as a neuropathologist, but as you can see from the MRI, there's very obviously a lesion right there, and that allows us to see the brain in a different way and see things that are not necessarily visible to the naked eye. It also allows us to essentially map the surfaces of the brain in a digital space that then allows us to have really specific neuroanatomic sampling and get really targeted areas when we do the microscopy. And finally, we can do volumetric analysis, measure how various regions of the brain have changed in regard to each other, in regard to the other side of the brain. So lots of really so this becomes an incredible resource, especially when used in conjunction with the microscopy. The way we do microscopy, this is something that a lot of brain banks focus on, is really moving towards a more digital approach. So right here in this top row, this is what a neuropathologist would see when a slide is under the microscope. We scan every single one of our slides from every case, and we're able to use artificial intelligence programs, digital pathology programs, to really dive into the next level. This is an overlay of that, one of those digital pathology programs. The program is called Halo. And as you can see, all of these little red spots and green spots are areas of interest for each of these different stains, each of these different slides. And using that image analysis, that digital image analysis, you're able to really take things to the next level as far as quantitation goes. You're able to count the number of spots, you're able to measure the size, and really explore this tissue in a way that's not possible just by looking at it under the microscope. Finally, the last part of the brain donation process is really the reporting. It's every single for every single brain donor we get, we write up a finalized neuropathology report. We send those to the clinical teams, to the research study teams, and to the next of kin if they are interested. We also, given, as I said, our focus on the fact that this should be a partnership, we invite the families and loved ones of our donors to come back and tour our lab, or have a sit down in a web conference with us to discuss our findings, ask questions. If they're in person, we're able to show them their loved ones' brains if they want, really just to make sure that they are part of our community and to know that, so that because we see this as a partnership and we want to help, we want to report back and to bring them in and make this a community effort and partnership. At this point, the next question is, how do labs get tissues? So studying human tissue poses unique challenges. There's a lot of, as I said, a lot of co-pathology. So I don't think it's really uncommon to get to look at a brain and see just one thing going on. So in the setting of Huntington disease, often we see the changes that are related to Huntington disease, but we also see a lot of other changes as well. We see age-related changes like Alzheimer's disease. We see strokes. We see trauma. So part of it. So this is where designing human studies is different than designing laboratory studies. There's limited availability of vulnerable brain regions. In Huntington disease, the areas that are affected, the basal ganglia, become really atrophied and are really limited. So there's also, it becomes a challenge to try to design a study using that tissue and maximize that resource for, to try to support lots of different studies. As a result of this, we have investigated, of these challenges, we have investigators fill out a requests form. This is the URL here. You can also find it on our website. I have a link to that at the end of this presentation. Part of this form is so that we can ensure that the brains that are donated to us go to reputable institutions, that they're not going to be used to do things like, they're not going to be sold for profit. They're actually going to be studied in scientific laboratories, in some cases used for drug development and therapeutic, and pharmaceutical companies trying to bring drugs to market. Also, this form initiates the material transfer agreement, so everything goes according to plan. We then invite investigators to come to a tissue committee meeting where we sit there and we just because it's easier to just hash out a lot of the details in person, we're not necessarily evaluating the merit of their science, but more we're trying to figure out how to best support them. So we're clarifying their research goals, defining the inclusion-exclusion criteria, and that allows us to help design the cohorts for their studies. And then it also allows us to establish relationships. So if they come back to us in 6 months or a year after doing these studies and say, we'd like to do further, it's really easy for us to jump in and know what they're looking for and how to get them more tissue. Finally, the cases and controls are selected by the brain lab team, neuropathologists, program coordinators, data managers, and then we send a list of candidate specimens to the investigator. And once everything's squared away, we package it and ship it. I'm not going to spend a whole lot of time going into this because I want to focus on the practical, but this is, there's a lot of really fascinating science that's coming out of what we're doing with digital pathology. This is some of the studies that we've looked at as we've talked about, as we're really interested in understanding why HD affects people differently, why some people have really prominent motor symptoms and others have more cognitive symptoms. So one of the things that we're interested in is looking at another protein called TDP-43, which is associated with other diseases like frontotemporal lobar degeneration, amyotrophic lateral sclerosis, and other age-related changes. And we wanted to know how this is impacting the brains of Huntington disease donors. So one of the projects we did was looking at going back to our repository, taking HD brains that went as far back as 1966 and evaluating the density of TDP-43 inclusions, how they were associating with Huntington, and whether there was a correlation with clinical course. And what we found was that in brains from our Huntington disease donors, they were 2.5 times more likely to have TDP-43 inclusions than those without. This is where you see that here. This is, you know, the HD brains with TDP-43 pathology, those without, versus controls with TDP-43 pathology and those without. And you can see that clear pattern here, that there's more, the HD, there's just more HD brains with TDP-43 pathology. We also found that it was more likely to be present in donors with older age and those who presented with cognitive or behavioral symptoms as opposed to motor symptoms. And the evidence that we found in the analysis we've done has suggested that it may exacerbate HD symptoms. So this is really going back to the archives, trying to figure out what, you know, or trying to learn from tissue that's been donated decades ago, and we're still able to learn new things. We're also able to use this for really cutting edge science and cutting edge analyses. This is an example of spatial molecular analysis, which is a cutting edge technique. Probably this is at the leading edge of current scientific analyses that allows us to see up to 6,000 genes on a subcellular level in a section of tissue. And we were able to take tissue from donors without HD, with HD, and with juvenile HD. And as you can see here, this is a digital rendering of what those are. And this is the control sample has this nice heterogeneous collection of cells versus Huntington disease, which has all of these blue cells here, which represent microglia, activated microglia. And interestingly, when you compare that to a juvenile onset Huntington disease, all of these yellow and reddish pinkish cells here, these are also microglia, but they have a completely different activation pattern. So again, learning brand new things from cutting edge techniques and really maximizing the donations that we've received. This is, we collaborate with HD laboratories worldwide. Some of the studies that we've been doing have been, they're pilot studies, and our collaborators have really taken them to the next level. And this map shows places that we've sent Huntington disease samples to in the last 3 years. And just in the last year, we've had a couple really phenomenal publications and preprints come out that have used our samples, used the brain tissue to make discoveries that would not have been possible in animal models. Tai Wen and Leslie Thompson at UC Irvine have looked at TDP-43 pathology, the protein I was just talking about. They've looked at it in a different way at the subcellular level and have made really groundbreaking discoveries, finding out that, finding patterns that nobody's ever seen before in Huntington disease brains and in any brain at all. So this is opening new avenues of science, opening up new areas of investigation. And then from a spatial standpoint, Sonia Vasquez Sanchez and Don Cleveland at the University of California, San Diego they released a preprint earlier this year. So this is available on bioRxiv that's really diving into spatial transcriptomic spatial analysis and showing and they have gone deep, and it is their work is absolutely beautiful, and I highly recommend digging into it, just because it's the discoveries that we're making with this tissue is really pushing the science to the next level. It's pushing therapies to the next level. So I hope that my enthusiasm for the research has showed you just how essential brains from HD donors are to really understand the biology, understand how it affects individual to individual, and how it's pushing our and opening the fields to new therapeutic options. So now the big question: how to get involved in brain donation? If you're interested in it, I want to start out by saying that, first of all, all brains associated with HD are incredibly valuable and incredibly appreciated. Whether you're an individual with HD or a family member of someone with HD, it's not, we would accept donations from anyone in the HD community just without hesitation. This is the best way to get involved with brain donation is to speak with your local HD clinical team. They're typically the ones who know our phone numbers, who know where the nearest biorepositories are. They often will have relationships with biorepositories. So talking to, if you're a patient at an HDSA Center of Excellence, speak with the coordinators or the neurologists there. If you're not involved with the Center of Excellence, talk with your neurologist, your psychiatrist, social worker, anyone associated with your clinical care will probably be able to connect you to a biorepository. Second of all, create a donation plan. Once you figure out a biorepository, discuss the plans with your family, with your loved ones, so they know what your wishes are. Make sure the consent forms are signed, and work with the biorepository for the logistics. The biorepositories will usually be able to help figure out things like transportation, figure out where, what the steps of donation are. Every biorepository is going to work in a slightly different way based on their geographic location and what resources are available there. But periodically confirm and update those plans with the biorepository. It can be really helpful in making sure everything goes smoothly. For questions regarding brain donation at University of Washington, this is our website here. This is our email address, our phone number. If you send an email to this, I'll be included on the email list. I'm part of this. I'll get those emails sent to this email address at uwnp@uw.edu. You can also email me personally. My email is online. You're able to find that. If you have any questions regarding donation or collaboration, feel free to reach out. And finally, thank you to all the brain donors. This is work that is just, all of this is because of brain donation that we're going to be able to understand HD, treat HD, and hopefully eventually cure HD.
That was fantastic. Thank you so much. And what an important message, right? I feel like so many of the major discoveries that we've had in the past 5 years have been because of human brain donation. So such an important message. We're running very short on time. So I guess I will just say for the Q&A, if you asked a question, I know Dirk was answering those live. So check the Q&A box to see if he answered your question. And then if you have questions and you weren't able to ask those, feel free to email me, sarahhernandez@hdfoundation.org, and I can help get those questions to Dan and Dirk, or Dan just very generously shared his email with everyone. So please feel free to use that. But with that said, we're going to wrap up there. I want to end by thanking everyone who donates to the HDF to support the research that we fund, and all of you for tuning in. And last but certainly not least, our speakers, Dirk, Dan, thank you so much for taking the time to tell us about the amazing work you're doing with human brain tissue. Our research webinar series will take a break for the summer, but we will be back in September. We'll be joined by Dr. Rachel Harding and Dr. Leora Fox from HD Buzz, who will bring us a breakdown of the 2026 HDF Scientific Conference that's happening this August. Registration for that webinar will be coming soon, so keep an eye out for that. Also, HD Buzz will be taking over HDF's Facebook page on Friday, May 29th, as part of our HD Awareness Month programming, answering any question you might have around research or clinical trial news. So mark your calendars and get your questions ready this Friday on Facebook from 8 a.m. to 4 p.m. We are so proud to showcase the advancements in HD research through our Research Spotlight webinar series. If you find these sessions valuable and you would like to support our mission, please consider donating today. Thank you for being a part of our community, and I hope you all have a fantastic summer. Thanks, guys.